All right. So my proposal is titled A Conformational Study to Determine the Effect of Ibrutinib on a Mutated BTK, PE24G, and PS21 using computational drug analysis and kinase inhibitor assay. So how does BTK work? So BTK is short for Bruton Tyrosine Kinase, and a mutated version causes lymphomas. So first, antigens, which are foreign body particles attached to BCR, which is B cell receptors. And B cells are cells that facilitate the immune system and helping the body fight diseases. So these antigens bind to BCR, which recruits CD79A and B, which are co-receptors. Those also recruit kinase LYN and SYK, which phosphorylates PI3K, and PI3K turns PIP2 to PIP3. PIP3 acts as a docking site for cytoplasmic kinase BTK and AKT, which phosphorylates PLC gamma 2, which activates PKC, and then that produces a set of secondary messengers that activate IKK. IKK phosphorylates I-kappa B, which the disruption of that leads to the release of NF-kappa B into the nucleus for cell proliferation, cell survival, and gene expression. So why is this important? According to the Cancer Association, more than 20,000 people die from lymphomas, and it is estimated that 900 people will die from lymphomas in the United States this year. So with the obvious problem being that nobody wants to be diagnosed with cancer or have their family members be diagnosed with cancer. Cancer is the second leading death worldwide. There's also a plethora of inhibitors that are being tested every day to find the cure of it. Traditional process is time consuming as well as risky and costly. So my research overview just focuses on these two mutations and to be able to determine which drug can be used to inhibit the mutation using the IC50 and to validate the result obtained from virtual screening by using wet lab experiment methods to determine the potency and specificity of the drug interactions and to also confirm the IC50 of the drug for each mutant. The importance of this study is to hopefully use computational methods to to reduce errors during experiment and to cut the cost in half. Virtual screening methodology. So by using BLAST and software such as PDB-Sum, we're able to find the protein's function, catalysis mechanism, as well as the properties of the proteins. And then protein preparation is done using Schrodinger software to remove any or modify unnecessary parts of the protein, such as hydrogen ions that could get in the way of the bonding. And then we have HCBS, which is used to kind of filter out the inhibitors and pick the top 10% hit each round. And then we have the induced fit docking, which is used to incorporate flexibility into the protein for more accurate representation of the protein complex and to dock the top 5 hits into the active site. Wet lab methodology. So as we did in wet lab, I'll be using kinase inhibitor assay. Kinase Glomax luminescent is a method used to determine the activity of the purified kinase by quantifying the amount of ATP remaining in the reaction. 5 tests will be done for each mutant, 1 blank, 1 positive control, and 3 concentrations of the inhibitor. Ibrutinib was my top inhibitor, so that's why I put it there. To determine the concentration of each inhibitor, unfortunately, we'll have to use the try and error method. Research plan and expected results. So first, to determine which compound is best to test, the analysis of literature value of the compound IC50 will be done. The IC50, if the IC50 is lower than the mutant compared to wild type, it is a promising drug to experiment on. Then kinase inhibitor assay will be done, as I previously mentioned, and then expected luminescent signal in a tube containing no inhibitor, no enzyme, just ATP should have the highest signal. Moreover, a tube that just contains the enzyme should have a low signal. Finally, a tube containing the highest amount of inhibitor, and the mutant should show high signal compared to a tube containing low amount of inhibitors. Originality and innovative perspective. This research again focuses on the verification of ibrutinib and other drug compounds to determine if they are substantial inhibitors for the mutant PS21 and PE24G. These findings can be used to then determine the specificity and potency of ibrutinib, and these are the compound from the obtained IC50 of BTK mutant. Work on these mutants have not been done before using virtual screening, and that's what makes a proposal unique. That's it.
Have to do the second round, so Mike, now your turn. Last question.
What type of cancer was it again?
Lymphomas.
Lymphomas.
Yes.
Again, where that tumor place in the body?
So it just says like chronic lymphoma leukemia. I think it's more of the B cells. So like B cells are used to facilitate the immune system and fighting off diseases. So B cells would have the tumor.
Okay, so title. You want change. If you want got A, you should change your title. Go back to your title. Say your title will confusion. Confirmational study determine effect of the FDA in future on mutant computation drug analysis. So you say this confusion, it's not the confirmation study. The study is to identify the novel inhibitor for this drug target to replace existing drug which shown the drug resistance.
Right.
So you want to rephrase your title.
Okay, I just figured that since the computational results show that ibrutinib is the top drug, like doing the wet lab would just confirm it or just, kind of show that it doesn't work. Like, so I just thought, but I'll change it.
So the ibrutinib, that's your top one compound?
Yes, that was the top compound.
Okay, so then this will very risky because you had 5 compounds testing. Now you see you want testing your top one compound. So you say you lose, you make your risky. And you can see the confirmation study to determine effect of the top 5 hit from virtual screening on that mutant.
Okay.
So change your title, make your title more safe.
Okay.
Okay, change the title. Okay, good.